So thanks again, Jill. This is actually my first eternacon, so really a pleasure to be here, and I'm really excited about all the talks this weekend. So looking forward to all that. So as Jill mentioned, I'm going to be talking about using Eterna in my second year biochem course, specifically supporting work integrated learning. So before I do that, I just want to acknowledge that at Capilano, our learning activities take place on the unceded and ancestral territories of the Lillooet. Lillooet, the Musqueam, the Sechelt, Squamish, and Tsleil-Waututh nations. So we're really privileged to be able to live and work and play here. So probably you aren't familiar with Capilano University. We're a small teaching-focused university located in Canada. So just to give you some context, we're up here in the Pacific Northwest area, a couple hours drive north of Seattle. So we have our main campus shown here on the right. Although we're in an urban area of a population of about 2.5 million people, we're really lucky to have our campus kind of nestled in the forest here with easy access to mountains and ocean and all these kinds of things. Are really a privilege to be able to work and play in this area. Okay, so again, the context here is work integrated learning. So maybe to give you a little bit of background on work integrated learning. So this, as defined by the International Journal of Work Integrated Learning, this is an educational approach that uses relevant work-based experiences to allow students to integrate theory with meaningful practice of work as an intentional component of the curriculum. And so just some examples of things that would constitute work integrated learning, things like applied research, apprenticeship, clinics, community service learning, co-op, internship, field placements, practicum, and work experience. So a key element of work integrated learning, so a couple of key elements. First of all, these initiatives require collaboration with an external partner. So it's not something you're doing just for the sake of a class internal to the class itself. And then secondly, the work that you're doing has to have value beyond the class. So this is a major focus of our provincial government in British Columbia. So we received some some provincial funding at Capilano University to try to build up our work integrated learning opportunities across our programs. Now, maybe in parallel to this, we've also been working in our Faculty of Arts and Sciences at Capilano in kind of reinventing our pedagogy according to what we call our academic model. So this is really a student-centered focus on pedagogy that's built on these four pillars of studio learning. So the idea of learning by doing, collaborative leadership. So not only working in teams, being a leader, but also things like working ethically, accountability, intrinsic motivation, communication, all these kinds of things. Innovation-enabled thinking. So this is the idea of getting students to be working on problems that actually have some relevance, again, beyond the classroom. And then the final piece, region integrated learning, trying to connect more effectively with our local communities. So of course, work integrated learning touches on really all four of these pillars. So given that we have this initiative to try to foster work integrated learning into our curriculum, the challenge for me was how can we incorporate work integrated learning into a second year biochemistry course? So we were specifically directed to try to integrate work integrated learning into both lower levels, so first and second year, as well as upper level courses. So some of the challenges in terms of incorporating work integrated learning, at the second year level, students really lack foundational knowledge and technical skills. And as a result of this, there's a big gap between what the students can do and what might be of value to a potential external partner. And then on top of this, at Capilano, as a small teaching-focused university, we're not a research university. We have limited laboratory resources. So this can, again, limit the kind of value of the work that we can do for an external partner. Maybe just give you some context on the course before we get into the specific project. So this is a second year biochem course. Many of you will probably recognize a lot of the elements of the curriculum. So about a third of the course is focused on protein structure, amino acids, primary, secondary, tertiary structure, folding, allosteric proteins, enzymes. We spend a relatively small part of the course on nucleic acid structure. So a lot of what Eterna focuses on really would be in that unit on nucleic acid structure. So I think that broadly speaking, and this is maybe well known to many on this call, RNA structure and function is still, I would say, underrepresented in the second year or in the introductory biochemistry curriculum. So I'm hoping as I develop this project through the course to try to expand out that component a bit. Okay, so now thinking about this work again, how to incorporate work integrated learning given the constraints that I mentioned. I mentioned that one of the elements for work integrated learning is applied research with an external partner. So I started thinking along the lines of maybe something computational based that wouldn't rely on laboratory resources. So I was actually aware of Foldit before starting on this project. And so I was aware of the citizen science element and had played it a bit myself. So I looked into this as a first option. It was really timely to have this talk preceding this one. Really intriguing. I'm really excited to see where Foldit has come and will be going in the next few years. At the time when I looked at this is kind of going back, I guess, a little bit pre-pandemic. At the time, it didn't seem to me that Foldit was really aligned with the kinds of things that I was trying to accomplish with this project. So in terms of, applied research, working with specific external partners, etc. However, it was through Foldit that I believe that I encountered Eterna, and I explored the platform. I hadn't heard of it before. I explored the platform, was really intrigued by many aspects, especially the labs that were sort of these discrete projects, applied research projects that were connected to specific research labs. So that seemed to fit the mandate of the work integrated learning. Also the opportunity for players to be involved in publication, etc. So it looked really exciting. I reached out to Riju. He very graciously was receptive to that connection, and he put me in touch with Slava Bukovic, who I think is actually on the call today. I saw her name earlier, as well as Jill. And so we started working together, trying to figure out some potential projects that we could work on through Eterna, putting me into contact with external labs, PIs, etc. So that was really exciting. So we started to develop that project over probably a year or so. And then I implemented it in the spring of 2022. So just to give you a bit of a sense of the project that we had come up with. So I've separated this into three parts. The first is a critical analysis of a paper in the literature relating to the project. So this is primarily coming from the lab of whatever the PI is that's on the particular project that we're working on for that term. So for many students, this is actually their first experience in reading a scientific paper, so it's a bit daunting for them. So the exercise is basically to analyze and summarize the paper and then post their analysis to a forum on the course website, and then students can basically read each other's summaries and analyses, post questions and comments, etc. So this is kind of giving us a little bit of a background or framing of the research project. So in parallel to doing this, they're then completing the Eterna tutorials, and then we move into the design phase. So we've done 2 iterations so far that I'll talk about. So a key element, again, for work integrated learning is working with external collaborators. So very importantly for this project, at the end of the term, after the students have completed their design project, they submit their designs to the collaborators. We've done it through Zoom the last couple of iterations, so that's been really great. It really helps to enhance the professionalism of the student presentations, etc. And then, of course, there's an element for some of these projects anyway to post the student designs to the Eterna page for voting and then potentially for investigation in the lab, which is also very exciting for them. So as I mentioned, we've done 2 iterations to date. The first one in spring of 2022 was a ribosome redesign project, but we'll talk about it in a few minutes. And then the second one, just this past spring, was the RNA pseudoknot project. So the ribosome design project, I'm sure this is very familiar to most of you on the call, but just for those of you who maybe don't come from this background, a little bit of a reminder of what the ribosome is and what it does. It plays a role in the information flow process, the central dogma of molecular biology, in basically reading mRNA and using that information to manufacture protein molecules. So we see the ribosome over here. Again, reading our genetic code, this is our mRNA, and generating our sequence of amino acids to form a protein that will eventually fold into a 3-dimensional structure. So here's the ribosome, kind of a little bit of an inside view. So we see that we have 2 broad subunits of the ribosome. The blue one is the so-called large subunit. The green is the small subunit. And within this structure, these yellow molecules represent tRNA molecules. So in this one, you can actually see the peptide bond between the 2 amino acids in the process of being formed. So the ribosome, part of the function is to bring these amino acids linked to the tRNAs in close proximity to allow the covalent bond to form between the amino acids. So the focus of this project was really on the large subunit. So actually, if I go back one sec, you might notice in our structure, we have sort of 2 different representations here. So we have these smaller, sort of darker purple structures in the top subunit, which are shown in darker green on the bottom subunit. So these are ribosomal proteins. And then in lighter colors, with the larger helices, these are the ribosomal RNA molecules. So looking specifically at the large subunit, we have two RNA molecules contributing to the structure here. So this is the secondary structure version over here on the left, folded into 3 dimensions, looks something like this. So you can see that most of the structure of the large subunit and the small subunit, for that matter, is actually comprised of the ribosomal RNA. And this large subunit contains the catalytic center that's involved in linking those amino acids. Now, normally, as you're probably aware, proteins are built from a standard set of amino acids. So it's 20, typically 20, or this particular image shows 21, including selenocysteine down here. So these are the building blocks that are used to manufacture proteins based on the mRNA sequence. So the focus of this project, so this was the Open Ribosome Evolution Round 1 project with the Jewett Lab at Northwestern University. So the focus of this project was to redesign the ribosome to allow it to accept modified amino acids. So for this project, the players, as they were designed, so maybe have participated in this one, the designs that players came up with were submitted to the community for voting, with the top 3 designs in each of the projects selected for investigation in the lab. So this, again, was really exciting for our students to be able to contribute to this kind of project. So a little bit more detail, we're looking at modifications to the 23S ribosomal RNA. So that's the larger RNA found in the large subunit to allow for protein synthesis with these non-standard beta cyclic amino acids. So typically proteins will use these so-called alpha amino acids, shown over here, where we have this amino group at the position adjacent to a carbonyl group. So we call this the alpha position right here. So alpha amino acids. These ones over here, these are beta amino acids. So we think about this carbonyl group, alpha, now beta amino, and then we have this cyclic component found in the amino acid as well. So these are unusual amino acids, typically not accepted by the ribosome, but these have potential applications in things like the design of therapeutics, design of proteins that are resistant to degradation, new material properties, etc. So I wanted to give you a little bit of a window into one of the student presentations that came from this project. So this was a group of 3 students in the class. So they were looking at, again, possible random mutations of the 23S ribosomal RNA of the ribosome that might allow it to accept these modified amino acids. So the approach that these students took, in fact, most of the students in the class took, they used a program, UCSF Chimera, which some of you may have used before. This is a molecular visualization software, freely available, allows you to look at primarily biological molecules in 3 dimensions. So this particular group, they were looking at the region of the ribosome involved in peptide bond formation. So given that these beta cyclic amino acids were a little bit larger, their rationale was, well, maybe we can look at modifying the amino acids that are kind of in the pocket surrounding the area where the peptide bond is formed, try to look at maybe we can make a larger space that might accommodate these larger amino acids. So this is just a small snippet. Most of the students in the class used similar kinds of approaches. Other students focused on different elements of the ribosome. For example, the exit tunnel, which is the area of the ribosome where the emerging peptide is released from the ribosome. So they really got into the molecular details for this project, which was really great to see. So that was the first iteration. The second iteration, again, focused on the pseudonaut design project. So this is with Riju Lab. And we primarily were connected with Rachel, his graduate student, and Jill was a great support for this in terms of defining the structure of the project, helping to put us together. This one, actually, although there was a pseudonaut lab, I believe, going on at the time, the timing didn't quite align with the timeline of our course. So Jill was really gracious in trying to put together a specific dedicated version for our course. So that was really appreciated. And so I'm thinking we're going to hear more about this pseudonaut project as the weekend progresses, but the goal here is to try to develop a large library of RNA sequences that could potentially form these pseudonaut structures. So just a little quick refresher on what a pseudonaut is. It's basically like a stem loop structure where we have the loop part now involved in base pairing with a separate part of the RNA molecule. And of course, these are getting a lot of attention recently. The COVID virus has been shown to have a pseudonaut in its genomic RNA that appears to be essential for viral replication. And so as such, it's a potential target for antiviral design. And it's been shown now that many other viruses have pseudonauts. So this could be a generalizable strategy for the development of antivirals. So I think we're, again, going to hear about this later on this weekend. However, the computational prediction of structures that can form or sequences that can form pseudoknots remains a challenge. So the idea with this project, again, is to try to develop pseudoknot structures that could then be tested in the lab. So from the students' perspective, what they were doing is they would use Eterna to design these pseudoknots. Jill very helpfully put together a tutorial video on how to go about doing that. Now, if you participated in the pseudonaut challenge, you may be aware that unlike other Eterna labs, there is no free energy reported by Eterna for these pseudonaut structures. But again, this kind of comes down to the limitations in prediction of pseudonaut structures from a computation perspective. So what the students did, Jill directed us to this other platform, Kinafold. So they were able to take their sequences that they generated in Eterna, bring them into Eternafold, and evaluate the stability of their pseudonaut structures. So the goal that I had set out for students was to basically try to come up with the most stable pseudonaut that they could through this kind of iterative process. So they design the pseudonaut, evaluate the stability, then go back in Eterna, refine the structure, optimize, bring it back in. And so again, the focus was to try to get the most stable structure possible, but also to develop a narrative. So like, what is the story that you're trying to tell? What's the process that you use to try to optimize your pseudonaut structure? Now, as they're developing these sequences, of course, they're submitting them to Eterna, where they'll be then used in testing, experimental testing, and that data will then be used to optimize Eterna folding engine. And again, I think we're going to hear more about that this weekend. So some of the student approaches, a little bit different from what we saw for the ribosome modification. So some students use, for example, an approach of optimizing GC content to direct specific regions of the RNA to pair with each other. Other students try to emulate natural pseudoknots, like the COVID one that I just mentioned. Others just use simple trial and error to see if they could just randomly change things around to come up with a pseudoknot. One student just basically tried to draw a pseudoknot, and then try to replicate that in Eterna. And then interestingly, this actually touches on something that Riju mentioned at the beginning in the introductory remarks. One student figured out a way to actually get ChatGPT to solve this problem, and was actually able to give it a command directing ChatGPT to generate 30 solutions for the pseudoknot challenge. So that's very intriguing. So this student's actually going to be continuing on with this work as a fourth year thesis project. So I'm really excited to see where that potentially goes. So just kind of sampling of some student results. So on the left, you see what they generated in Eterna. This is the same structure now in Kinafold. So it actually highlights some of the potential issues that we have here in computational modeling of these pseudoknot structures. You see that the one on the left has one pseudoknot, as shown by these lines indicating pairing between these two regions, whereas the structure generated by Kinafold actually reveals two potential pseudoknot regions. So again, some further understanding required to better understand how these things fold. So just to summarize, I want to finish off with summarizing with some comments. So this is a comment from Slava, commenting on the students who did fantastic. This is the first iteration involving the ribosome. So pointing out the complexity of the ribosome and the quality of the work. So I agree that was really great to see. I think that presenting to the external collaborators was very helpful in sort of raising the level, more so than if they were just presenting to me. So that was really great to see. Comment from Jill for the second round of presentations. So they were less in depth than the 2022 presentations on the ribosome, which I completely agree with. So looking at particularly the approaches that were used by the students into the pseudoknot challenge didn't really get into a lot of the atomic level investigations. They weren't really looking at the fundamental biochemistry. They kind of used these different approaches to coming up with these pseudoknot structures, but they weren't really grounded in the sort of structure function side of things that I would have liked to see. So hopefully we can refine that for future products. Again, another one from Jill, just emphasizing that both these iterations encourage students to think through a real world scientific challenge, formulate a solution or investigation approach, and recognize the types of challenges that researchers encounter. So I agree with all of that. I think it was effective in sort of giving students a first taste of what research is like. So just some successes, again, gives the students a research experience, kind of helps them to figure out whether that's something they want to pursue as a career down the road. It also encourages scientific creativity in a way that maybe isn't as accessible in a conventional teaching approach. As you saw with the pseudoknot challenge, students were able to use very different approaches to solving the same kind of problem. Again, the projects are focused on team-based collaboration, working in either groups of 2 or 3. So they had to work collaboratively in that context. And then, of course, as I mentioned, the engagement with the external collaborators was really positive to see. Some of the challenges in terms of implementing this project within the course, those of you who take in second year biochem or teach second year biochem will know that typically this is a very content-heavy course, and as a result, there's a lot of time that we have to spend just on the core curriculum of the course, and that means there's little class time that's available for the project. So the students are mostly working on this kind of in parallel to the course, as opposed to us working together in the classroom to try to solve these problems. Another challenge that Jill was very graciously able to help us with was the coordination of the timelines for, say, the Eterna labs with our course schedule. So they don't necessarily always align, but again, which was very helpful in setting up a specific version of the Pseudonaut challenge for us that would work with our timeline. And then finally, this one maybe for the players seems trivial, but in order to access the labs, of course, students first need to complete the Eterna tutorials. Now, for students who are, have a full course load, are very busy, some of them found this a bit frustrating because it does take several hours of work, and some of those tutorials, of course, can be quite challenging and frustrating. So that was one potential hurdle for student engagement with the project. Now, in terms of the next steps, what I'd like to do is try to strengthen the connections to the class content. As I mentioned, currently RNA structure is really only a small part of our curriculum. I'm looking at ways of expanding that to better integrate the Eterna project into the class. It'd be great to explore potentially custom design of projects that would better align with the course content. So I'll hopefully be in touch with Jill and others about that in the future. I'd really like to increase the experiential element, and so specifically I'm thinking about integrating a laboratory component. So because of that, I'm especially looking forward to the next talk in the conference today, looking at ways of potentially integrating some lab work into the project. Also looking at extending the projects beyond the course. So right now the students are working over a single semester on this project. I mentioned one student is now extending this into their 4th year thesis project. I'm looking at maybe ways of building these projects into over multiple courses. For example, maybe the course that I'm talking about right now is not a lab course. We'd have to kind of do the lab work within the normal scheduled hours. But is there a potential, for example, to work on sort of the game-based, the computational-based side of the project in one course, and then maybe extend the activities to a subsequent course? And then finally, although I've spoken to the students about this project kind of informally, we'd kind of debrief, I'd like to integrate a formal student reflection component to this piece to really dig into their thoughts and how they feel it impacted their learning, how they feel it may potentially impact their possible choice of a career in science and specifically research. So I'll just wrap up with some acknowledgements. So thanks especially to Jill and Slava, who were my main contacts through Eterna and helped to sort of set up these projects. Really, really grateful to both of you for that. The first project, the ribosome project, was done in collaboration with the Jewett Lab at Northwestern. So I especially appreciate Dr. Andrzej Kruger, who participated in the student presentations. Really great to have that involvement. And then the second project was with Reju's Lab, in particular working with Rachel, his graduate student. Again, she attended the presentations, was really great to have her insight and input. And then finally, Dr. Sarah Smaga from the National Science Foundation Center for Genetically Coded Materials. Sarah also attended this last round of presentations for the Pseudonaut Challenge and gave some really helpful recommendations and advice in terms of future direction for this project as well. So with that, I'll wrap up and happy to answer any questions anybody might have.